Hi, folks. My name is Carra Putri Ghaisani, and people usually call me Carra. So, today I would like to talk about a topic that lies at the intersection of science, ethics, and also humanity. So, should human gene editing be allowed? While science asks what we can do, society must ask what we should do. So, through this presentation, I will explain what human gene editing is, what are the benefits, and I would like to examine the ethical concerns, and also we will discuss whether it should be allowed under strict conditions. So, by the end of this presentation, I hope to provide a balanced perspective on this important and also controversial issue. So, let me introduce you guys to human gene editing. So, what is human gene editing? Human gene editing is a scientific process that allows scientists to change, remove, or even add specific DNA sequences in human cells. But actually, gene editing itself is not only applicable for humans, but it could also be eligible for plants, animals, and other living stuffs. But since we're talking about human gene editing, so I will discuss about human. So the goal of this human gene editing often to correct genetic mutations that usually cause disease. So there are two types of human gene editing. The first one is somatic gene editing, and the second one is germline gene editing. First, somatic gene editing. Somatic gene editing usually targets non-reproductive cells such as blood or even skin cells. The changes only affect the patient of being treated and are not passed on to the future generations. So, because of this, somatic gene editing is generally considered more ethical and is already being used in lots of medical research. For example, the treatment of genetic disease like sickle cell disease, and we will talk about this later. And on the other hand, there is germline gene editing, which is usually targets embryos, eggs, or even sperm cells. And any genetic changes made at this stage will be inherited by future generations. So this type of gene editing is much more controversial because it raises serious ethical, religious, and also safety concerns, including the lack of consent from future individuals. So in conclusion, while somatic gene editing is often supported for some medical purposes, germline gene editing remains highly debated and is mostly restricted worldwide. So right now, we're gonna talk about the scientific promise. So one major benefit is the prevention of serious genetic disease. Because by correcting some faulty genes, scientists usually may be able to stop the inherited conditions before they cause a lifelong suffering. And then another important advantage is the improvement of quality of life. Because gene editing, especially the somatic gene editing, can help patients live a healthier life without even passing genetic changes to the future generations. And then, human gene editing also contributes to the advancement of science and also medicine because it could allow researchers to better understand genetic diseases and develop more effective treatments. And in addition, gene editing can be seen as an extension of medical care. Usually it could be similar to other accepted medical technologies such as gene therapy and also organ transplant. And finally, by preventing genetic disease, gene editing has the potential to reduce long-term healthcare burdens, both for families and even for healthcare systems. So, despite about its scientific potential, human gene editing also raises a serious ethical concerns. So one major issue is a safety risks and unintended consequences. What does this mean? Because gene editing technologies may cause unexpected genetic changes, the long-term effects are still not fully understood. Another concern is about the lack of consent, especially in germline gene editing, because future generations cannot agree to genetic changes that will permanently affect their lives. And then there is also the risk of designer babies. So where gene editing could be used for like a non-medical purposes, such as enhancing appearance or even intelligence, which may increase like a social inequality. And additionally, many people raised the playing God argument because believing that altering human genes like crosses moral or religious boundaries by interfering with natural or even divine creation. And then finally, there is a strong moral responsibility to future generations because since genetic changes can be inherited, society must consider whether it is ethical to make irreversible decisions without even knowing their long-term impact. So right now, I will give a real-life example like about treating a sickle cell disease with CRISPR-Cas9. So, what scientists did, doctors used CRISPR-Cas9 gene editing on the patient's own bone marrow cells because bone marrow cells are like the source of red blood cells. So after the bone marrow cells are edited, the cells were returned to the patient's body, and the treatment itself like correct genetic problem that causes a sickle-shaped red blood cells. And then the real outcome, the patients who received the therapy stopped experiencing severe pain crisis, and many no longer required regular blood transfusions. And also, in recent clinical trials, patients usually like showed a dramatic improvement in quality of life. So, in conclusion, human gene editing, it could present like a both great promise and also serious challenges. So therefore, human gene editing should not be completely banned, in my opinion, but it must be strictly regulated, limited to medical purposes, and also guided by ethical responsibility. And ultimately, the debate over human gene editing reminds us that scientific progress must always be balanced with moral values and respect for human dignity. So that's all for today. Thank you so much for listening. Bye bye.